Hello everyone, welcome to the new session of Genetics and Plant Breeding, Cell and its Structure. So this is the second part. We are going to study about the cell and its structure. Cell envelope and its modification. So prokaryotic cell, most prokaryotic cell, particularly the bacterial cell, and have a chemically complex cell envelope. So this cell envelope is present where? It is present in the bacterial cell. So what does this cell envelope composed of? Cell envelope is made up of three different layers. What are those layers? The outermost glycocalyx. The second layer is cell wall. And the third layer is cell membrane or plasma membrane. Now this unitedly make cell envelope. And this is present in the prokaryotic cell. So the cell envelope consists of tightly bound three-layered structure. That is glycocalyx, cell wall, and the plasma membrane. So the cell envelope is made up of glycocalyx, cell wall, and plasma membrane. Okay, so here we can see this structure that plasma membrane is there, cell wall is there, and capsule is there. So we will study about the capsule. Actually, the outer layer can present in the form of slime layer or capsule. Right? So although each layer of the envelope perform the distinct function, they act together as a single protective unit. So this cell envelope provide protection. What is it is meant for? It is meant for protection to the cell. So bacteria can be classified into two groups. They can be classified into two groups. What are the groups? Those groups are gram-positive and gram-negative bacteria. Okay, so they are treated with a stain. They are treated with the strain gram, and if they take those gram stain, they are called as gram-positive bacteria, and the other that do not take gram stain, then they are called as gram-negative bacteria. So the one which take the stain because they have a peculiar complex structure of their cell envelope, and the one which do not take, it have a different from the gram-positive cell envelope structure. Okay, so on the basis of this staining, the bacteria are classified as a gram-positive and gram-negative bacteria. Glycocalyx differ in the composition and thickness among the different bacteria. This is very, very important. So glycocalyx are not unanimous in all the type of bacteria. Their composition and thickness is different in different bacteria. So it can be loose sheath. It can be in the form of loose sheath, and that is called as slime layer. Very simple. Slime layer means a thinner layer of this glycocalyx is present in some, while other it may be thick and tough and form a capsule structure. So when this layer glycocalyx is a very loose and thin sheath, it is called as slime layer, and when it is thick and tough, it is called as capsule. Okay, so the glycocalyx is different in different types of bacteria. Cell wall determines the shape of the cell and provide a strong structural support to prevent the bacterium from the bursting or collapsing. So this is the main organelle that provide the shape to the bacteria, right? And it also helps them from bursting or collapsing. The plasma membrane is a selective permeable in nature and interact with the outside world. So there is a 3 different layer, that is the glycocalyx. Glycocalyx is also present in the 2 form, that is it can be present in the slime layer as well as in the form of capsule. Okay, next comes the cell wall, and the innermost layer of the cell envelope is the plasma membrane, that is selectively permeable one. This membrane is similar structure to that of the eukaryote. So cell membrane or the plasma membrane is the membrane which is very much like present in the case of eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of the plasma membrane into the cell. Okay, so mesosome is present somewhere, like plasma membrane get invaginated, and it forms a structure which is called as mesosome. Okay, so it is compared with the mitochondria in the case of eukaryotes. Why? Because the enzymes which are present in the mitochondria are found on the surface of the mesosome. So the mesosome is nothing but it's an extension of the plasma membrane, invaginate itself and form a coil structure which is called as mesosome. These extensions are in the form of vesicles, tubules and lamellae. So this mesosome can be in the form of vesicles, tubules or lamellae. They help in cell wall formation, DNA replication and the distribution of daughter cell and the respiration and secretion. So they also help in DNA replication because DNA once get attached here and it helps in the process of replication. It also helps in the process of cell wall formation, right? So this mesosome structure is very, very important. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm and that are called as chromatophores, which contain pigments. Okay, so these chromatophores are nothing but the extensions of the membrane, which is found in the case of cyanobacteria, and they contain pigments. So these pigments are for process of photosynthesis. They support the process of photosynthesis, right? So cyanobacteria do the process of photosynthesis. Bacterial cell may be motile or non-motile. They can be motile or non-motile depending on what are the motile structure they are having. If motile, so suppose if the bacteria is motile, then they have a thin filamentous extension from their cell wall, which is called as flagella. So suppose this is a bacteria, so if it is a motile, then it will have a flagella-like structure like this, flagella-like structure. So this is a flagella. It helps in locomotion of bacteria. So bacteria show a range in the number and arrangement of flagella. Flagellum is composed of 3 different parts: filament, hook and basal body. So if this is a bacteria, so here it will have a basal body, here it will have a hook and it will form a filament like structure. So this is a filament, this is a basal body. and a hook like structure, right? Beside flagella, pili and the fimbriae are also surface structures of the bacteria. They are attached to the surface structure. So beside flagella, they are having pili and fimbriae, right? The pili are elongated tubular structure made up of a special protein. So they are, if this is a bacteria, so they form a tubular structure and they get attached with another bacteria. So sometime in the process of conjugation, that is the sexual reproduction, in the case of bacteria, they transfer their genetic material with this tube-like structure, which is called as pili, right? So this is its function. Fimbriae, they are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rock in streams and also to the host tissue. So fimbriae are basically for the attachment, right? Cell membrane. So here we have talked about the cell envelope. Now we are going to discuss about the cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. So when the electron microscope was discovered, then only the detailed structure of the cell membrane or the plasma membrane was studied. Okay. Meanwhile, the chemical studies on the cell membrane, especially in the human red blood cells, that is RBC, enabled the scientists to deduce the possible structure of the plasma membrane. The first time the structure of the cell membrane was revealed by studying human red blood cell. Okay. So these studies shows that the cell membrane is mainly composed of lipids and proteins. So what are the constituents of the cell membrane? Cell membrane constituents that are protein and lipids. Okay, so the major lipids are phospholipid. So what type of lipid is there? Lipids with having phosphate group attached to it. That is why it is called as phospholipid. Example, lecithin, that are arranged in a bilayer. So how this phospholipid look like? It look like this. Bilayer. So bilayer lipid structure like this. So this is hydrophilic and this is hydrophobic. So this, if we study about this, one of the lipid molecule of this membrane, then it is having a head and a tail like structure. So head structure is hydrophilic. It loves water, right? So that is why it got its name hydrophilic. And the one which get away from the water is called as hydrophobic. So this is the bilayer arrangement of the phospholipid in the cell membrane. See, how it is arranged. That is why it is called as bilayer, right? Lipids are arranged within the membrane with the polar heads towards the outer side and the hydrophobic tails toward the inner part. So we have just seen the structure of the lipid bilayer. This ensures that the non-polar tail of the saturated hydrocarbons is protected from the aqueous environment. Aqueous environment means the one which is having liquid or the water, presence of water. So this polar head, its outer side means it is hydrophilic, and the one that is hydrophobic tail, that is present inner side. Okay. In addition to the phospholipid membrane also contain cholesterol. So some cholesterol is also present on the phospholipids. Later, biochemical investigation clearly revealed that the cell membrane also possesses protein and some carbohydrate structure also. So beside lipid and protein, some carbohydrate is also attached to the cell membrane. The ratio of protein and lipid varies considerably in the different cell type. Okay, so it is not that is a fixed that the lipid percentage is this much and protein percentage is this much. It is not fixed. It varies considerably according to the type of cell. But in human beings, the membrane of the erythrocyte has approximately 50 to 52% protein and 40% of the lipids. Okay. Depending on the ease of extraction, membrane proteins can be classified as an integral and the peripheral protein. So, if we draw a structure of a cell membrane, so we can see two types of protein. So, this is the lipid bilayer. Okay. This is a protein. This is a protein. And we can see some extrinsic protein. Okay, this is also protein. But the one which is present in between the lipid bilayer, this is lipid bilayer, lipid bilayer. So this protein is present in between the lipid bilayer. So that is called as integral protein, integral protein. Okay, and the one which is present at the outer surface only, then it is called as peripheral protein, peripheral protein. So peripheral protein lies on the surface of the membrane, while integral proteins are present partially or totally buried in the membrane. So we have seen just now that how the integral protein and peripheral proteins look like. So here we can see a diagram for the cell membrane. So here you can see this is the integral protein structure, and here you can see the phospholipid bilayer. This is the bilayer, right? And here you can see some peripheral proteins are also attached on the surface, peripheral protein, right? And at the peripheral protein, this structure you see here is the sugar, that is carbohydrates. So three different structure make, or three different biomolecules makes your cell membrane, that is protein, lipid and carbohydrates. Okay, some amount of cholesterols are also attached to the surface. An improved model of the structure of the cell membrane was proposed by Singer and Nicholson in 1972 that was widely accepted as a fluid mosaic model. So this was the improved model of the structure of the cell membrane. So it was proposed by Singer and Nicholson, which is very famously known as fluid mosaic model, right? According to this, the quasi-fluid nature of the lipid enables lateral movement of the proteins within the overall bilayer. So protein is moving, continuously moving, okay? Because this lipid is a fluid-like structure. So when lipid moves along with the fluid, the protein also move along with it. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions. Okay, cell and cells are connected, right? So they forms the formation of intercellular junctions, secretions, endocytosis, and cell division, right? So this fluidity in the present in the cell membrane helps in so many ways in the cell. One of the most important function of the plasma membrane is the transport of the molecules across it. So this is very, very important, said that certain structure has to enter inside the cell, and also some of the structure that has to be withdrawn out from the cell. So this fluid mosaic model or this fluid structure of the cell membrane helps in that ways. The membrane is selectively permeable membrane. This membrane is selectively permeable. It allows only certain molecules to enter inside the cell, right? So to some molecules present on either side of it, it is selectively permeable. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Okay, no requirement of energy. Some molecules can enter inside the cell, that is called as passive transport. Neutral solutes may move across the membrane by the process of simple diffusion, that is higher concentration to the lower concentration, that is according to the concentration gradient. Concentration gradient, that is from higher concentration to the lower concentration, but that molecule must be neutral. That must be neutral, right? Water may also move across this membrane from higher to the lower concentration. Movement of water by diffusion is called as also osmosis, right? So water moves from high concentration to the lower concentration through the semi-permeable membrane. Semi-permeable membrane. That is from higher concentration to the lower concentration, right? So this is called as osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. So some of the intrinsic protein that are present in the cell membrane function as a carrier protein. So they helps to allow this non-polar lipid bilayer, okay, to some of the polar molecules. They allow some of the polar molecules to enter inside the cell through this carrier protein. Few ions or molecules are transported across the cell membrane against their concentration gradient, that is from the lower to the higher concentration. Such transport is an energy dependent process. So now here it is moving from lower concentration to the higher concentration, right? With the expense of energy, that is ATP, and it is utilized. So ATP is utilized in such a transference or transportation of molecules across the membrane. Okay, so that is called as active transport. An example is sodium potassium pump, right? Cell wall. A non-living rigid structure called cell wall forms an outer covering for the plasma membrane in case of fungi and plants. So in the plant cell, we have seen that the outer layer, outer rigid structure, right, is made up of cell wall, right? So it is not gives a shape to the cell, but also protect the cell from the mechanical damage and infection. So beside giving the shape to the cell, it helps the cell, or it protects the cell from mechanical damage and infection. It also helps cell to cell interaction and provide barrier to undesirable molecules. So undesirable molecules are not allowed to pass through the cell wall, right? Algae have a cell wall made up of cellulose, very important. Cellulose, galactans, mannans, and minerals like calcium carbonate. While other plants, it consists of cellulose, hemicellulose, pectins, and some proteins also. But the basic component is the cellulose in both the case, right? But here in the case of algae, we can see that they are also having, beside cellulose, they are having galactans and mannans, and some of the minerals like calcium carbonate. But in the case of plants, they are having cellulose, hemicellulose, pectins, and proteins. So here we can see the structure of the cell wall. The cell wall of a young plant cell, the primary wall is capable of growth. So here we can see in the middle structure, this is middle lamella. Middle structure is called as middle lamella. And we are having primary wall, this black color, primary wall. Primary wall, right? So this primary wall is capable of growth, which gradually diminishes as the cell matures. So when cell get matures, this primary wall cannot be seen, right? Secondary wall is formed on the inner towards the membrane side of the cell. So we are having primary wall, then we are having the layers of, we can see here, layers of secondary wall toward the inner side, that is towards the membrane. Okay. The middle lamella is a layer mainly composed of calcium pectate, which holds or glues the different neighboring cell together. So this is a mineral, calcium pectate, present in the middle lamella. The cell wall and the middle lamella may be traversed by the plasmodesmata, which connect the cytoplasm of the neighboring cell. So we are having some gap in between the cell wall. Those gaps are called as plasmodesmata. So it forming the cytoplasmic connection between the one cell to the another cell. Okay. So they are traversed. The cell wall and the middle lamella may be traversed by the plasmodesmata, means that is the connection between the two cells. So this is all about the cell wall structure. So in this session we have studied about the cell envelope, plasma membrane, as well as the cell wall. Cell envelope is present in the case of bacterial cell or the prokaryotic cell, whereas the cell wall is present only in the case of plant cell as well as the bacterial cell, that is the prokaryotic cell, right? Cell wall is present in the animal cell, plant cell, as well as the prokaryotic cell, means eukaryotic as well as the prokaryotic cell, right? So this is the boundary. Today we have studied about the boundary of both prokaryotic and the eukaryotic cell. So in the next session, we are going to study about the different organelles present in the prokaryotes and eukaryotes. Till then, thank you very much.